It's now my pleasure to introduce tonight's speaker. Dr. Craig Cole is a board-certified hematologist at the University of Michigan Comprehensive Cancer Center in Ann Arbor. He's currently an assistant professor, Division of Hematology-Oncology, focusing on multiple myeloma, and is committed to patient education. In 2016, Dr. Cole became a member of the IMF's prestigious International Myeloma Working Group, and we're happy to have him with us. And we're pleased to have Dr. Cole present tonight on our call and to also field your questions at the end of this presentation. Please remember, if you have a question, to type it in under the control panel under questions. And so now it's my pleasure to turn tonight's call over to Dr. Cole.
Well, thanks, Robin. Welcome everybody, and huge thanks to Robin and everyone at the IMF for inviting me to talk about a couple really exciting things, a couple of the new treatments and novel therapies in myeloma, as well as clinical trials. So what we will talk about tonight is first we're going to just get some definitions out of the way, especially in regards to how we actually see if these new therapies actually work or not. And so it's good to get everyone on the same page regarding what is it exactly as a response with myeloma. The second thing that we'll talk about are then the novel agents, and two in particular that are really exciting, the BCL2 inhibitors and the checkpoint inhibitors. We'll talk about how those work. And then finally, we will talk about clinical trials, because there are a lot of myths about clinical trials, and we hope to dispel some of those. One thing that sometimes, to get everyone on the same page, one thing that we need to talk about just and how responses work in myeloma is that people will have a certain number of myeloma cells in their body, and then that equals the M protein in the blood test. So the bone marrow has plasma cells in it, and we can measure those with just a blood test with the M protein. And when someone receives a therapy for myeloma, then that causes the number of plasma cells to go down. And instead of repeating a bone marrow biopsy, the only thing we have to do is just recheck a blood test, and we can see that the M protein goes down. And so how we actually tell if a therapy worked or not is using the IMWG response criteria. And a complete response is when there's only 5% cells left in bone marrow. But really, the primary way is that we don't see any evidence of any protein in the blood test or in the urine. A very good partial response is when there's a 90% decrease in the blood protein level or in the urine. Partial response is a 50% decrease, and stable disease means that the disease hasn't progressed at all and has remained the same. And then how we actually tell if a drug has worked is we look for an overall response. So how many patients did respond to a clinical trial drug with at least a partial response or 50% decrease in their protein. And then there's something called the clinical benefit rate, which isn't a formal part of the response criteria, but is basically, how many patients had a benefit from being on a particular therapy with stable disease or better. And then to see how long a particular therapy lasted, we look at overall survival, and then probably more, or really importantly, and probably our main test the litmus test for therapy is the progression-free survival. So how long from the start of a new treatment until its failure? How long did that particular treatment last for people? And then finally, AEs, adverse events, so we'll talk a bit about, and those are the side effects of a particular trial drug. So I hope that's kind of clear about the responses. And there, and try to to start with some of the old therapies of myeloma. Before we had a lot of science about myeloma, we used non-targeted therapies. So we used chemotherapies and steroids when we didn't have any idea about how the disease worked. And thank goodness that over the past 20 years, there has been a lot of laboratory testing that's been done in order to have us understand the complex biology of how myeloma works, what makes up the plasma cells, what surface proteins do they have on them, and exactly how do they work inside the bone marrow with the bone marrow stromal cells. And based on a lot of that science, there have been drugs that have been utilized in clinical trials to actually address a lot of that biology. The proteasome, of course, is what is targeted by the proteasome inhibitors like Velcade, Kyprolis, and Marrow. The Emmit therapies attack multiple areas on the plasma cell and the bone marrow stromal cell, with Polymus being the most recent one of those. And then there are some of the new drugs, Faradac, and the drugs that are currently in clinical trials. And then there are drugs that attack the antibodies, or the antibody drugs, which attack the surface of the plasma cell, including the recently approved daratumumab and recently approved elotuzumab. And those drugs have really improved the response rate, especially when combined with the IMiDs and the proteasome inhibitors. And then some of the more recent new drugs attacking the biology of myeloma, actually the checkpoint inhibitors that we'll talk about later on, that actually gets the T cells to engage the plasma cells. So all of this new biology in myeloma has given us a wealth of targeted therapies that are infinitely better than using old-fashioned chemotherapy. So, and of course, I didn't expect anyone to, remember all those pathways. But now we'll talk about how we've actually applied that science of all those pathways that I showed in that slide to the novel agents in multiple myeloma. And so here's a plasma cell, and this plasma cell has a receptor on it. And cytokines are growth hormones that help myeloma proliferate. And when those cytokines engage their receptor on myeloma cells, it activates several pathways inside several reactions, several wires inside the plasma cells, which activate the myeloma cell nucleus to do certain things. And a lot of those pathways have called JAK2 and STAT pathways, RAF/MAP kinase pathways, and the PK3 pathways. But basically, all those pathways activate the machinery inside the bone marrow, inside the plasma cell nucleus, to produce proteins which in part help keep the plasma cell alive, so pro-survival proteins. Proteins that help keep the plasma cell from, allow the plasma cell to reproduce, so those proteins which help myeloma cell reproduction. And then there are additional proteins that are set up by these pathways, which allow for other supportive cytokines to then feed back in to the pathway to have the cell proliferate and be resistant to cell death. And of course, having cells proliferate without dying is kind of the heart of any type of cancer. And so there are other pathways that actually go through a protein called BCL2. And BCL2 is a protein that's outside the nucleus. And what it really does is it works through those pathways originating at the cytokines, and really concentrates on anti-cell death and pro-survival. So BCL2 really works on having the myeloma cell stay alive primarily, and not die in the face of steroids or chemotherapy. So if we look at, and the cells that, the myeloma cells that really do use that BCL2 are the patients that have the chromosomes 11 and 14 bound to each other. So they're translocated. We have 2 copies of all these chromosomes in our body. But in myeloma, when the mutation happens where the 11th chromosome and the 14th chromosome stick together, they end up causing a gigantic increase in BCL2 inside the plasma cell. So when the cytokines engage through these different pathways, when patients have the 11-14 translocation, BCL2 is turned on in a major way. And when BCL2 is turned on in a major way, then we get lots and lots of proteins that are anti-cell death and pro-survival proteins. And so those patients that have 11-14 will have lots of pro-survival proteins made, and then they'll have lots of plasma cells that are reproduced but never die, and they're immortalized by BCL2 and these pro-survival proteins. Now, a smart thing to do would be if we had a way to stop BCL2, if we had a way to inhibit BCL2, and we didn't have BCL2 there, then those different signaling pathways would not activate BCL2. There wouldn't be any pro-survival proteins made by the nucleus, and then the plasma cells would disappear, and then the myeloma could be treated. And when the myeloma is treated, then people are happy, and that's a happy face. So I hope that that made sense. And what we can now do is apply, so I'm sure it's clear as mud, but we will apply that BCL2 to a drug called Venetoclax for relapsed myeloma. And this was a trial that was presented at the American Society of Hematology meeting this last December. And we just talked about how BCL2 enhances the survival mechanism of cancer cells and allows them to be resistant to chemotherapy. And there's a pill that was made called Venetoclax that is a BCL2 inhibitor, which allows cells to die and treat cancer. And that was approved by the FDA in 2015 to treat a specific type of cancer called CLL, not myeloma, but for chronic lymphocytic leukemia. And in chronic lymphocytic leukemia, it was able to do to treat the highest risk CLL very well, and it had some side effects causing low white blood counts, nausea, and anemia. But one major side effect was that it was so powerful in getting rid of the cancer cells that there would be a sudden release of electrolytes into the body called tumor lysis syndrome. But otherwise it was and that was a rare occurrence, but otherwise it was a very safe drug. And so there are laboratories done specifically on Venetoclax for multiple myeloma cell line samples. And it was found that, just like I had shown in the cartoon, that those patients that had the translocation of 11-14 were especially susceptible to cell death with Venetoclax. And so cell cultures and cell lines that had 11-14 really in the laboratory, the drug really worked in the laboratory. And especially in those cell lines which overexpress that BCL2, that was the main protein to induce the plasma cells to stay alive and not die. And based on that laboratory data and that the drug was approved for CLL, we had some idea what the safety is. There was a clinical trial done specifically on patients with multiple myeloma looking to see how Venetoclax worked in myeloma to see if there would be responses. And so this study was done primarily at the Mayo Clinic. And they enrolled 66 patients. And what they did is that it was a phase 1 study. So they increased the dose slowly to find out what was the dose that had good responses and if it had any side effects. The really interesting thing about this clinical trial is that it was done without any dexamethasone. So one thing thing we would love to do with myeloma treatment is divorce ourselves away from the steroids. And so in this clinical trial, the Venetoclax was used as a single agent, and Dexamethasone was only included on patients who didn't respond to therapy. So when the study was first opened and we looked at the responses, it was only a 21% overall response rate. And if we said, gosh, that's not such a great response rate for patients with relapsed myeloma. However, if you looked at just the patients who had the 1114 translocation, that was 30 patients, the response rates went up significantly in those patients that had the 1114 translocation. Again, this is a BCL2 inhibitor. Patients with translocation 1114 have lots of BCL2, and so we would expect them to do especially well. The patients that didn't have didn't have 1114 didn't respond so well to this therapy, but if you look at the patients that had high expression of that BCL2, like I showed in that cartoon, that the 1114 caused a high expression of BCL2, that resulted in remarkable response rates in just this patient population. And those with even low expression of BCL2 were not much different than the patients in the overall population. And so what that means is that in patients that were treated, had previously treated multiple myeloma and had multiple prior therapies, that this drug was safe and worked as solo therapy for myeloma. The 600 milligrams was the high dose, and it caused some abdominal pain and nausea. Thank goodness there was no tumor lysis syndrome found like it was seen in the other types of cancer. The overall response rate, people would say, was pretty okay at 21%. But if you look at just at the patients who had 1114, then the response rate doubled. And when you look at the response rate in those patients that had the BCL2 overexpressed, then the results were remarkable. And the reason that I'm so excited about this is that this is the beginning of precision therapy for multiple myeloma. And usually what we do is that people come in, a patient comes in, and we give them all the same therapy. And some of them will benefit, some of them won't benefit, and some people have adverse events. Now we have precision medicine is when we do a test to see, will this therapy work in this patient? And we'll give them drug therapy specific to their abnormality. So we look at to see how many patients have the BC. We only give this drug, Venetoclax, to just the patients have the BCL2 and the translocation. That makes them happy because they have a better chance at responding, and the other patients don't need. We're not going to waste their time in a drug that doesn't work as well with them. So this is precision therapy just for patients that have the translocation, and it's the beginning of other therapies that will be in the future that will attack specific drug targets, specific genetic targets in multiple myeloma. And that, I think, is a very exciting future for myeloma. So the next thing I wanted to talk about is talking about the checkpoint inhibitors, the B cells and T cells. So how can we get those guys engaged in helping, how can we get your own immune system engaged in fighting multiple myeloma? So we'll talk a little bit about how checkpoint inhibitors work. So the immune system is an incredible part of our bodies, and it has the ability to tell normal cells from abnormal cells. So you want your immune system to be able to tell the good guys from the bad guys, and the immune system can do that by using checkpoints. So what it does, the immune system uses checkpoints which are molecules on cells to be able to tell the abnormal cells who need to be attacked and the normal cells that need to be left alone. And so our immune systems are able to tell bacteria or cancer cells and tell them and distinguish them from normal cells like the eyeball cells and the heart cells. And that's kind of the miracle of our immune systems. Cancer cells, though, have found ways to avoid the checkpoints and avoid being attacked by the immune system. And that is what we have now turned that around to try and get the immune system to attack cancer. So here's, let me show you a cartoon of how the checkpoint works. So here's a T cell, and the T cell is the green guy at the top. And what T cells want to do is they want to attack and to remove abnormal cells from the body. And so what these T cells will do is that they have a protein on them called PD-L1. And PD-L1 fits inside, it's the checkpoint, and it fits on a protein on cells called PD-L1. So PD-1, if it meets PD-L1 on a cell, then the checkpoint, the ID check works perfectly, and the cell is identified as a good guy cell. So the T cells check out cells. If the PD-1 fits with the PD-L1, so if you go to if you go to the bar and they will check your ID, if the ID checks out, you get to go into the bar if you're over the age of 21. And if you're a good guy cell, the T cell will check your ID, the PD-L1, and if that fits out, then you're a good guy and you avoid being attacked by the immune system. And the good guy cells live very well. However, if you're a bad cell and when the T cells come to attack, if you don't have any ID if you're a bad cell and you don't have any ID, then you're identified as being a bad guy cell. And the absence of having PD-L1, if you don't have any PD-L1, then you must be a bad guy, and then you end up being destroyed by the immune system. So if I go to I go to a bar, and if I don't have any ID, then they'll throw me out. If a bad guy cell doesn't have PD-L1, if it doesn't have ID, then the immune system attacks, and then the cell dies. And that is how our bodies are able to distinguish bad guy cells from good guy cells and able to destroy bad cells. However, multiple myeloma has figured out how to fake an ID. So it has a fake PD-L1 on its surface. So even though the T cell knows that this may be a bad cell, when it actually tries to engage the multiple myeloma cell, when it goes down to check it out, it finds out that the PD-L1 and the PD-1 fit together perfectly, that the ID checks out. Even though the T cell knows this is a bad guy, the ID checks out and has to leave that cell alone. So PD-1 and PD-L1 fit perfectly, and even though it's a bad guy, the immune system ends up leaving it alone. A bad guy disguised as a good guy, and the immune system leaves it alone. And which is a lot like disguising yourself or the myeloma cells disguise themselves as a wolf in sheep's clothing. That the myeloma cell has a protection and is able to fake its bad, evil status from the T cells. So what if we could change the ID? What if we could either remove PD-1 from the T cells, allowing them to go forth and destroy the plasma cells as they would want to, or what if we could have an antibody against PD-L1 which would then remove the ID, remove the sheep's clothing off the wolf, so the immune system can find the cell and attack it. So if you can remove either the PD-1 or PD-L1, if you remove the checkpoint from the either from the cancer cell or the immune cell, then the T cell can do what it wants to do, which is attack the bad cells. Your own immune system attacks the cells, sees a bad guy as a bad cell, and then the bad guy cell dies, as well as the T cells actually call in for backup, and more of the T cells engage the bad guy cell. Now that the ID of the bad guy cell is seen as bad, the immune system fully attacks the cancer cell. And that's what we want to have happen using these drugs, the anti-PD-1 and anti-PD-L1. So that, I think, is and it uses your own immune system. It uses your own immune system to have and decloaks the plasma cells from the immune system ignoring it. These checkpoint inhibitors have already been approved for different types of cancer. So atezolizumab has been approved for bladder cancer already by the FDA and has undergone scrutiny with clinical trials to make sure it's safe and effective. PD-1 antibodies have been approved by the FDA. Nivolumab and Pembro have been approved for melanoma and lung cancer. And at the last ASCO meeting, the American Society of Clinical Oncology meeting, there was a clinical trial called Keynote that was specifically for multiple myeloma using Pembro, which is one of the PD-1 antibodies, plus Revlimid in patients that had been refractory or relapsed from Revlimid already. And the response rate in using these checkpoint inhibitors, so just utilizing the immune system in patients that have already failed Revlimid, that there is an overall a 50% response rate in patients that had relapsed refractory myeloma and using this brand new technology, the checkpoint inhibitors. Even patients who had failed Revlimid, were failing Revlimid at the time they were entered on the study, still had a 38% response rate, which means that it was just the checkpoint inhibitor that was making that 38% response rate, because these patients had already failed Revlimid. And 88% showed some decrease in their protein at baseline. So there was which is really exciting, and there was a study that was just discussed at the last American Society of Hematology meeting in December, where patients that had relapsed and refractory myeloma were given Pembro, the PD-1 inhibitor, plus pomalidomide and dexamethasone. And people who responded after 24 months remained on maintenance doses of Pembro and pomalidomide. And these patients had at least 3 prior therapies, and some of them had 5 prior therapies, and all these patients have been relapsed and refractory to the usual therapies that we use to treat relapsed, newly diagnosed and relapsed myeloma. So these patients had failed other therapies. And when you look at the side effects, the side effect profile was of some fatigue, neutropenia, low platelet counts, and a lot of these you would expect sometimes with the pomalidomide, but there are very few high-grade side effects with this therapy. The one question that is, I think, very important is, well, what if you have these T cells that no longer have any inhibition to destroying cells? Will they, by mistake, attack normal cells? So if we remove the checkpoint, what would keep those T cells from, by mistake, identifying normal cells? And those are the immune reactions at the bottom of the slide. That there were some patients, 10%, that had some level of low thyroid or pneumonitis means some lung inflammation. So there was in this study some side effects that included the immune system being actually overactive and causing some difficulty with the lungs and with the thyroid. It also caused some vitiligo, which when the melanin cells in the skin disappear because of the immune system. And so there were some crossover effects, but for its ability to treat myeloma, in this population that had multiple prior therapies, the response rate was very good at 65%, and the clinical benefit of this combination keeping patients stable was 72%. And the primary thing that this therapy was able to do is it was worked across all risk groups. That high risk groups, the refractory patients, and the full population had very much the same number of responses, and that's actually very exciting for our patients that have high risk disease. But a lot of the responses end up being stable disease. Or partial responses in a therapy that utilizes the immune system. So the median duration of response has been about 16 months, but the overall survival of this population hasn't been reached yet. So patients are still receiving the Pembro, pomalidomide, and Dex, and they're still surviving their disease while on this therapy. And so side effects did occur in 50%, but it was very rare for patients to have to discontinue therapy, and most of the side effects were very manageable. And this is just the tip of the iceberg. There are several clinical trials going on using the Emmit drugs like pomalidomide, Revlimid, the antibody drugs such as daratumumab, and including those with the checkpoint inhibitors to really stimulate the immune system to destroy cancer cells. And so there are a lot of clinical trials that are opening utilizing this technology. And what I just showed you is just the tip of the iceberg of these very exciting therapies in multiple myeloma. So are clinical trials good for people? Yes, they are good for people. And so we're going to talk a bit about clinical trials now. And so what was the world like before we had clinical trials? Well, one of the first therapies for multiple myeloma was urethane. And urethane was a weed killer and a chemical weapon. And it was thought that maybe that would work for some blood disorders. And so there was a paper that was published using only 2 patients in 1947. That wasn't that long ago. And those 2 patients that this weed killer was used on, they felt that it worked. And only based on 2 patients that urethane became the standard care, the standard treatment for multiple myeloma for 20 years without any clinical trial. And the only proof that we had that these drugs worked was in 2 people. So finally, in 1966, there was a randomized trial that was done to see if urethane really should be the standard of care. And it was randomized between Coca-Cola, and this isn't a plug for Coca-Cola, that you could have used any type of soda, but Coca-Cola was patients received either Coca-Cola or urethane. And what happened? There was no difference in survival. Not because Coca-Cola was especially good, but it was because urethane didn't work at all. And so therefore we entered the days of clinical trials to have proof that drugs actually work for patients instead of giving people weed killer and not knowing if it worked or not. So clinical trials are great. And so how does medical care advance? Well, we start out with therapies that are bad and when we have little technology, we have started with therapies that were bad, like urethane, that had poor responses. So someone did a clinical trial with urethane versus Coca-Cola, and then the standard of care at least went up by removing urethane. But each time that there's a clinical trial that's done, it improves this and becomes the standard of care. And what happens is that as clinical trials come out and new therapies become the standard of care, we get better and better at treating myeloma. That the toxicities gets less and less, the effectiveness gets greater and greater, and it becomes inevitable that as we do more clinical trials and we improve the standard of care, that eventually we have to get to the point of a cure because of the velocity of improving the standard of care. And we've been doing this for the past 20 years, and we're getting closer and closer to improving the standard of care to the point of a cure. So where does the standard of care come from? It comes from clinical trials and the participation of patients working with doctors to make things better for everybody. So what are clinical trials? Well, clinical trials are carefully controlled research studies, and they are conducted by doctors at various centers to improve the cancer treatment in patients. The reason we do clinical trials is to study a new therapy and compare it to the standard of care, and therefore if the therapy works well, the old standard of care is removed, and the new standard of care becomes the previous clinical trial. And that's how things get better and better. With each clinical trial, there's a very detailed description of how the trial works and exactly what does it do, and that's a written protocol. It's what drugs are being tested, who's eligible for the clinical trial, and where the clinical trial program is taking place. And how drugs develop, and we saw a little bit of this earlier, is that initially drugs are tested in the lab, then are tested on animals. Then the FDA, the government has to be involved to see, is there enough evidence to prove that this clinical trial is safe for humans? So there are no one's a guinea pig in clinical trials. We look to see, the FDA has to approve the clinical trials for human use before it even comes near anybody. And the phase 1 clinical trial is the first clinical trial, so it's called phase 1, and it looks to see what's the safety of a particular drug, and what's the dosage, and what's the best way to give the treatment. Again, the FDA has already approved that it's safe to give at all, and now we look to see, well, what are the side effects? If that study works, then a phase 2 study is done, and it looks to see, well, how well does it work? Let's see, how well does it treat cancer? And that's usually the really big studies. And then the biggest studies are the phase 3 studies. And that's when you compare the old treatment to the new treatment. And it sometimes takes thousands of patients to be involved in those trials. And if it shows that the new treatment is better, the FDA approves it, and then it becomes a new standard of care. Phase 4 is when the drug becomes available early for patients to be able to have access to. One thing that gives people some anxiety is the idea of randomization. I kind of wanted to address that right now. Many clinical trials are randomized, and cancer clinical trials are randomized between new treatments and the standard of care. So you have patients, a number of patients, and when they're randomized, the computer generates, subdivides the patients into either a control group receiving the best available treatment, the standard of care, so no one gets a placebo. Patients with cancer get the best care available, or they are in a study group which gets the best care available plus a study drug, or they get just a study drug. So no one gets kind of gypped in the process, that when you're randomized, you get the best treatment available, or you get the best treatment available plus the standard of care drug, which means that everyone is happy, both the patients in the control group and the investigational group. Everyone's happy because they received the best care. So randomization is not, no one gets a placebo with that. So in facts or myths, if people will say, and if I enter a clinical trial, I'm going to end up getting a placebo. And that is a myth, that we don't, for cancer, we don't use placebos in clinical trials. Unless the standard of care is doing nothing, which is very rare and is very, very, very rare, multiple myeloma, placebos aren't used in clinical trials. You have to receive some type of standard of care treatment or the new treatment. Another myth is that a clinical trial is a treatment of last resort, and that's not true anymore. That clinical trials are for patients at every stage of disease. They're clinical trials for smoldering myeloma, for relapsed refractory myeloma, newly diagnosed myeloma. There are even some clinical trials monitoring patients with MGUS, and so they're trials at every stage of disease. Clinical trials are riskier than FDA-approved drugs. Well, that's a myth. In fact, the rigorous process to get a clinical trial started in the first place really assures that the drug is safe, and it's been tested a number of times before the FDA will even allow the clinical trial to be done. So it has a good chance of success by the time it gets to being part of a clinical trial. And some people say, well, if my doctor doesn't mention clinical trials, it must not be right for me. And these days, there are so many clinical trials, and doctors and patients have so much information that your doctor may not remember that there's a clinical trial for you. And so you need to be empowered to be able to ask your doctor, is there a clinical trial for me? Can you look up a clinical trial for me? Or or ask your doctor, is there a myeloma doctor or specialist that you could ask about a clinical trial? And some people think that the clinical trial is more important than the patient, and that is a myth. I really wanted to dispel that the main reason we do clinical trials is to help people. And the primary star of the show of a clinical trial is the patient. And if you don't like the clinical trial, if you're having bad side effects or if there's something you don't like about it, you can stop at any time. A clinical trial isn't a marriage contract. You're not marrying anybody when you get on a clinical trial, and you can leave at any point. And the big myth that has to be dispelled is if I in a clinical trial, I'll be a guinea pig. And I hope I've shown you already that there are no guinea pigs in multiple myeloma clinical trials, that by the time that the trial gets to you, it has been thoroughly investigated and safe. A clinical trial will not turn you into a guinea pig, I can assure you. So clinical trial patients, because of the close monitoring that's required, they get a lot of attention. They get a clinical coordinator, they get a research coordinator, and they have their nurse helping them through the process of receiving the clinical trial drug. And the safety is closely monitored in clinical trials, and many centers will have several different review boards to go through the clinical trial to make sure it's safe, including the main one, the Institutional Review Board, which is a committee of experts making sure that the clinical trial is ethical, safe, legal, that there's very little risk, and that privacy is protected for patients. Anytime that anyone's involved in a clinical trial, your doctor must give you a form called an informed consent document. And you have to read that and sign that before you enter a clinical trial. And that is an important document because it explains everything that you should go through in the clinical trial. But you need to make sure that you ask questions if you don't understand, and have your doctor go through the clinical trial with you if you have any questions. And make sure that you bring an advocate. It's always good to have a second set of ears to listen, especially when talking about clinical trials. And take your time in reading a clinical trial. There should be no pressure to enter a clinical trial. And there are so many clinical trial opportunities. The list that I have here is really inadequate for the number of clinical trials in multiple myeloma. There are new proteasome inhibitors being explored. There are new Emmit drugs related to Revlimid, Revlimid, and pomalidomide being explored. There are a number of monoclonal antibodies, checkpoint inhibitors, and a plethora of small molecules, including those which are targeted therapies for specific patient groups, that precision medicine that I talked about. And there's a trial for just the high-risk 17p mutated patients. So I hope that it's clear that clinical trials are an important option for everyone, that they can be for newly diagnosed patients, those with limited disease, advanced disease. We have worked really hard at having clinical trial opportunities for everyone at every stage of disease, and we need to make sure that we include patients of different ages, genders, and races to make sure that these drugs actually work in the American population. It is critical that people of ethnicity and patients that are older are involved in clinical trials to know that the treatment really works for the population. You need to take into account these factors and the other treatments used to get the right therapy and the right clinical trial. And remember that that communication is the key to entering a clinical trial. Make sure that you understand everything about it, that you've had all your questions answered, and that you are the star of the show. You are the empowered person in multiple myeloma treatment in clinical trials. Multiple myeloma treatment in general, but especially clinical trials. How do you find clinical trials near you? Where there's the IMF, and this is fantastic. I just used this, I tested it myself to make sure it works before I had mentioned it, but it definitely works. So if you go to the IMF website and you go under Advancing Treatment, there'll be a tab for clinical trials, and that will lead you to the IMS myeloma matrix, where you put in your zip code and your stage of disease, and it will give you all the clinical trials in your area. I would be remiss if I didn't mention the government's clinical trial engine at cancer.gov. And there's a big purple, if you go to this website, the government's website for clinical trials, there's a big purple button that you hit, and it will lead you to the clinical trials in your area also. And that's it. So thank you for your time and attention.
Well, Dr. Cole, that was fantastic. So take a sip of water, because I think we'll go right into some questions for you. And everyone on the phone, I can tell you that this myeloma matrix really is super helpful. I use it all the time, not only for patients that ask me questions when I'm visiting support groups, but also for myself, so just to stay up to date on what's happening and where. So Dr. Cole, I wanted to ask you a little bit more about the monoclonal antibodies. There's been a lot of excitement in that, and I've also noticed that we've got some questions in particular to Darzalex. Can you speak to that a little bit, please?
Oh, yes. So one of the just remarkable breakthroughs that have happened in the past, just in the past couple of years in myeloma therapy has been the monoclonal antibodies. And so those antibodies basically attach to a protein on the plasma cell. So every cell in your body has proteins which identify it as a heart cell or eyeball cell. Plasma cells, and especially myeloma cells, have very specific surface proteins which identify them as cancer cells. And unfortunately, people don't make antibodies to myeloma. But in the laboratory, they've made antibodies to multiple myeloma that can harvest the power of the immune system and direct it straight at the myeloma cell. And so drugs like Darzalex and the other monoclonal antibodies are very specific targeted therapies for the surface proteins on myeloma cells, and it directs your own immune system to attack it. Those drugs worked very well as solo agents, but the recent FDA approval was for Darzalex to be combined with Revlimid and Dexamethasone, or Darzalex to be combined with Velcade and Dexamethasone. And those two clinical trials showed fantastic response rates for the combination of the antibodies directing the immune system towards the myeloma cells and the standard therapies like Revlimid and Velcade to enhance that effect to treat myeloma. So those have been really breakthrough drugs in myeloma, and really, again, the tip of the iceberg in myeloma treatment, especially in harvesting the power of the immune system to destroy cancer cells. So, yep, those are the Darzalex and Elo are really, again, breakthrough drugs in myeloma therapy.
Okay, excellent. So now we're going to go to questions. So people, please remember, if you have a question, it's easy enough to just type it over there in the question box. We've got a bunch to go through already. And the first one comes from Bill. And he says, what are the most promising new therapies for relapsed myeloma that are close to being approved?
So I think that we've already, had the approval of Feradak in 2015, the histone deacetylase inhibitor, and Darzalex, and the recent approval for Darzalex combined with Revlimid and Darzalex and Velcade, and elotuzumab being approved. So there are other drugs that are in phase 3 trials right now, including new drugs in the updated version, a new histone deacetylase inhibitor, a new version of Feradak, and other combinations of antibodies with new drugs that are in phase 3 trials now to see how well they work. And so those are some of the approaching drugs. Any clinical trial that is a phase 3 trial is really looking to see, is it better than the standard of care, and can that drug replace the current standard of care? And so there are the histone deacetylase inhibitors, there are other antibodies that are in the pipeline, and the combination of the current antibodies with new drugs, including using carfilzomib with other antibody drugs. The ones, so those are all coming down the pipeline for relapsed patients. There's also drugs that are being looked at specifically for patients that are relapsed, having multiple prior therapies. And I didn't have enough time to talk about some of the one of the new drugs that is a inhibitor of the proteins of nuclear export, which is a very targeted therapy for myeloma called selinexor, and that is one drug that's being pursued specifically for the relapsed patients, especially after having relapses after the antibody therapies and some of the newer therapies. And gosh, I could talk about this all day. And the other really exciting thing is that there are clinical trials taking place right now on the combination of the checkpoint inhibitors that I talked about and the antibodies. And that is incredibly exciting because that superpowers the immune system. The combination of the antibodies and the checkpoint inhibitors superpowers the immune system to target myeloma cells. And I think that there was a lot of excitement about those clinical trials opening, including that we are hoping that these will be able to reduce the use of dexamethasone.
Okay, that's great. And again, if people go on to the Myeloma Matrix 2.0 and they just click on phase 3 trials, and then you can see on the screen that we're looking at, you can select trials for newly diagnosed, smoldering, maintenance, or relapsed refractory, or all. So it really is a good way to find out what those trials are and where they are and what the qualifications are for them. So we're going to move on to our next question now from Carl. And Carl is asking, does the acceleration of treatment options diminish the value of a stem cell transplant?
Wow, that's a very good question. So what we would what we'd like to do and right now the stem cell transplant is a very important part of cancer treatment. And I think that for myeloma treatment, and I think that stem cell transplant will always be a tool in the tool belt for us to use. And so is the value of clinical trials going to diminish, is new clinical trials going to diminish the value of transplant? Transplants are always going to be part of the tool belt, but we want to actually surpass a transplant. The whole point of doing clinical trials is to improve the standard of care. So sitting right now in 2017, where transplant is currently an option, I do sincerely believe that in the next few years, that as the clinical trials go on and we get more and more smarter therapy, specific therapy with less side effects, we have to get rid of the older therapies. So at some point, will transplant go to the wayside? At some point, it will always be part of the tool belt, but as the standard of care gets replaced by better therapies, it is going to be a therapy of the past. Just like some of the other therapies that we had. We used to have chemotherapy, Velcade, vincristine, Adriamycin, Dexamethasone. That was a standard of care, 15 years ago. That's been replaced by new therapies. And transplant today is going to be replaced by new therapies in the future. So it doesn't diminish it as much as it will always be there as part of the treatment algorithm, but it has to be replaced by something superior.
Great. And it's always really wonderful that we've got all these options today. So I think that transplant is still around for sure. The next question is from Georgia, and it's an interesting question. She said, I'm doing well on my current treatment. Would you suggest I enter a clinical trial to see if I can even do better?
That is a very good question, and the majority of clinical trials are looking are really focusing on a change. A change in what the disease is doing. So a change which means either things are doing well, that you've gone from you've recovered from a stem cell transplant, and that the change is to enter a maintenance therapy. So there are trials for maintenance therapy, or there are trials for newly diagnosed. So when you're newly diagnosed, of course, that's a change, and there are therapies available, or clinical trials available in relapsed refractory. And so it's always a good idea, I think, to be prepared. And so to have the conversation with your doctor, if something were to change with my disease, is there a clinical trial that we could think about in the future that you know about? And so if you're doing well on a therapy now and the side effects are minimal and everyone's happy, then I would stay on that therapy. But I'd always be prepared and looking and having the conversation with your doctor about, and healthcare team about, are there clinical trials available if something were to change?
All right, so I think we're going to do maybe one or two more questions, and I just want to let everyone know, if you didn't get your question answered tonight by Dr. Cole, please don't forget we have the IMF info line that's always available for you, and I will give you that information at the end of this call. So we have another question from Jeanette, and she wants to know, are the BCL2 drugs effective for patients with T414 cytogenetics?
T414. So that's again, every question is really good. I wish I could just, if my wife would let me, I would sit here all night and just answer questions. So there was another clinical trial that was discussed at the American Society of Hematology meeting, where they had combined the BCL2 inhibitors, Venetoclax, with Revlimid. And there are a number of clinical trials. Now that we see that Venetoclax works, the question is can it work for other populations besides the translocation 11-14? And actually in a study that was done in France, that combining Venetoclax with Velcade actually does have very good response rates, especially in patients with without a lot of prior therapies, and that would and the response rates were a bit higher for those that were 11-14, but across the board on that particular study, the combination of Venetoclax and Velcade worked for a number of different myeloma subtypes, and so the answer would be yes, for combinations of Venetoclax. The really interesting thing is that for specifically for those that are 11-14, that the using Venetoclax alone is also very effective. So combinations in other groups works, and solo therapy in the 11-14 group works.
All right, so I think the last call we're going to take tonight, and then again, I will give everyone the IMF info line number to call if you didn't have your question answered. The last call, or the last question, is about the Pollux and Castor trials, which Dr. Dury speaks of often. And so if you cannot tolerate Revlimid but want to be on the Pollux protocol, can you try pomalidomide?
That's, again, a very good question. And so the Pollux and Castor trial were trials and it actually led to FDA approval of utilizing utilizing Darzalex, Revlimid, and Dex, or using Darzalex, Velcade, and Dex. And so and that combination worked very, very well for patients. But they have been studies looking at other combinations with Darzalex, and specifically there have been studies, and there are ongoing studies utilizing Darzalex pomalidomide and Dexamethasone, and there will soon open a clinical trial around the country utilizing Darzalex, pomalidomide, carfilzomib, and Dexamethasone. So are there other combinations? I should say that there are other clinical trials utilizing a number of the other therapies with Darzalex, and of course, the first approval for Darzalex was with Darzalex therapy alone, which did have a 30+ % response rate. So is there more therapies beyond the Castor and Pollux trial that were, so revolutionary? Absolutely. And there are clinical trials, and there is data already out about the use of Darzalex plus other therapies. So yes, that's available, and there's much more to come.
All right, I love the ending. Much more to come. That's a good thing in the myeloma world. So the IMF, we all know, is a wealth of information, and I hope everyone knows that the website you should go to is www.myeloma.org. And I also want to make sure that you know that the replay of this teleconference will be available maybe as soon as tomorrow. So you can access all of the IMF previous teleconferences and this teleconference on our website by going to myeloma.org and then clicking on IMF TV. I want to also just give you a couple of important reminders. The IMF is going to be holding a patient and family seminar in Short Hills, New Jersey on April 7th and 8th. If you live in the Virginia Beach area, the IMF is holding a free regional community workshop on April 1st. So please call or go online to register for either of these programs, as they're really filling up quickly. And March is Myeloma Action Month, so please make sure that each of us take actions. It really does matter, and you can find more details on all the ways you can help, again, on the wonderful IMF website. And last but not least, don't forget that if you have more questions, the IMF info line has the answers. 1-800-452-CURE, which is 2873. Missy, Paul, and Judy are available to take your calls Monday through Thursday from 9 a.m. to 4 p.m. Pacific, and Friday is 9 a.m. to 2 p.m. Pacific. And once again, Dr. Cole, this was really wonderful, very informative. Your slides were fantastic. So thank you so much for taking the time tonight to talk with us. And again, thanks to our sponsors, Celgene and Janssen. And most importantly, thanks to all our participants tonight on this call. I hope you've learned and you do well. Have a wonderful evening, everyone. Thank you.